My topic is chemical basis of muscle contraction. What are muscles? Muscles are biological machines that are made up of proteins, converts the chemical energy from the ATP and CP. They are able to take, receive and respond to stimuli. We can only control voluntary muscles or skeleton muscles. Types of muscles: skeleton muscle, cardiac muscle, smooth muscle. First one, skeletal muscle. Skeletal muscle is a muscle tissue that is attached to the bones and is involved in the functioning of different parts of the body. These muscles are also called as voluntary muscles as they come under the control of the nervous system in the body. And this is the anatomy of skeletal muscles. Muscle proteins and chemicals. First one, actin. Actin is a family of globular multifunctional proteins that form microfilaments in the cytoskeleton and the thin filament in muscle fibrils. It is found in essentially all eukaryotic cells where it may be present at the concentration of over 100 μm. Its mass is roughly 42 Dalton with a diameter of 4 to 7 nanometer. Myosin. Myosin are motor proteins that interact with actin filaments and couple hydrolysis of ATP to conformational changes that results in the movement of myosin and the actin filament relative to each other. During each cycle, myosin moves 5 to 25 nanometer and one ATP is hydrolyzed. Tropomyosin. Tropomyosin are the large family of integral compound of actin filaments that play a critical role in regulating the function of actin filaments in both muscle and non-muscle cells. These proteins consist of rod-shaped, coiled hetero or homodimers that lie along alpha helical groove of most actin filament. Sarcomere. Sarcomere is a basic contractile unit for both striated and the cardiac muscle and is made up of complex mesh of thick filaments, thin filaments, and the giant protein titan. Troponins. Troponins are the group of protein found in the skeletal and cardiac muscle fibers that regulate muscular contraction. Troponin tests measure the level of cardiac specific troponin in the blood to help detect heart injury. Muscle fiber regions. A muscle sarcomere's border are defined as a Z disc or Z line. The borders of a single sarcomere are marked by two neighboring Z discs along the microfibers. The Z discs are where the thin filaments are attached. The eye band is a thin filament rich area of a striated muscle sarcomere. When examined under a light or electron microscope, this area is closer to the Z disc and is the lightest portion of the sarcomere. The thin filaments are the only ones in the eye band. The H zone is located in the center of a A band where the thick and thin filaments do not overlap. As a muscle contracts and the sarcomere shortens, the H zone shrinks. The M line. The M line is the attachment point for the thick filament in a striated muscle sarcomeres. The M line runs through the middle of the A band and therefore through the sarcomere. The A band is a myosin thin filament containing portion of a striated muscle sarcomere. In reality, the A band extends the whole length of the sarcomere's thick filament. It measures about 1 meter in length. Process of muscle contraction. Sliding filament theory is a popular theory proposed by A.F. Huxley and H.E. Huxley, also known as ratchet theory or walk-along theory or modern theory of muscle contraction. The very first process of muscle contraction it starts in brain. The nerves carries a signal in the form of impulse of potential difference and lead to the neuromuscular junction which is nearly touching the muscle cell that but separated by synaptic cleft. While muscles have junctional fold with postsynaptic membrane. There are two space, extracellular space, intercellular space. In extracellular space, which leads to the membrane potential in sodium and potassium ions, which leads to the depolarization and creates a chain reaction and further increasing the difference. In intracellular space, once the particle level, that is action potential, is reached, an enzyme released to degrade the ACH, preventing further sodium ions to entering. But the action potential continues to journey to T tubules which leads to the calcium channels. The action potential then causes the calcium levels to rise in the muscle cells. The calcium ions then binds with the troponin, and this leads to change in the shape in which binding sites available. Now the myosin head will pivot and bend, pulling the actin filament along and using ATP in the process, and the muscle contracts. Muscular change during contraction. Muscular change during relaxation. Binding Ca plus 2 ions to troponin. Ca plus ions separated from actin filament by sarcoplasmic reticulum. Troponin. Ca2 plus complex removes tropomyosin blockage of actin sites. In this, Ca2 plus returns to the sarcoplasmic reticulum. Ca2 plus released from troponin. Ca2 plus complex. In this head of myosin, ATP complex form cross bridge to actin filament. In this, troponin permits tropomyosin return to blocking position. In contraction, hydrolysis of ATP includes conformational changes in the heads of myosin. In this relaxation, myosin actin cross bridge breaks. In contraction, 1 mole of actin plus 3 moles of myosin actomyosin. In this relaxation, ATP myosin complex reformed in heads of thick filaments. Thank you.